you so much, Carol. Okay, well, thank you so much to Carol and Ellen, really, for the opportunity to talk with all of you today about an overview of approaches for developing therapeutics. I just want to make sure that this deck is really just used for informational purposes and not for medical decisions. So I just briefly wanted to introduce myself. I'm currently an independent consultant, so I focus on research and drug discovery. And this is just a timeline of experiences that have shaped me and guided me through my career. So I grew up in the Midwest, loving Girl Scouts and going to the zoo. So I learned a lot about caring for others and the biology of animals, so both of which have guided me throughout my life. I put gold stars on the experiences that have been the most powerful motivators in my career. And these are really when I had the opportunity to interact with patients and families and contribute to therapeutic development. So in my early 20s, I began working at a company focused on neurodegenerative diseases called Elan Pharmaceuticals. And I had the opportunity to go on rounds with a neurologist. His name was Dr. Miller, and I got to watch him as he went through and talked with his frontotemporal dementia patients. And I learned pretty dramatically and was confronted by the realization that there were very few options available to patients for interventional treatment. This really ignited my passion to understand mechanisms involved in diseases of the brain and to devote my life to developing therapeutics to improve patients' lives. So to that end, while I was at Ionis Pharmaceuticals, I was fortunate to work on a team that spanned multiple companies, including Ionis and Biogen, that identified a compound or an antisense oligonucleotide called Qalsody, and that has now been approved for SOD1-mediated ALS. And then while I was at the N. Lauren Foundation, an organization dedicated to developing RNA-targeted therapeutics for N of 1 patients, was so fortunate to work with so many patients and families, learning their stories and working to develop therapies to improve the lives for patients who had no help or hope. So today I'm super excited to talk with you about therapeutic approaches that are being used to develop therapies for ultra-rare diseases. So first I'll give an introduction to all the modalities, and then I wanted to share with you Susanna's story, which is very personal to me as I worked with her and her family while at N. Lauren to develop her personalized treatment. It's a multifaceted, complex process to develop an N of 1 experimental therapy, so I then wanted to talk through that process with you. I then wanted to share the history of therapeutic development for orphan diseases, and following with a comparison of the process between traditional approval and N of 1 therapies. And then finally, I will walk through companies that are dedicated to developing therapeutics for ultra-rare diseases. So I put up this slide just as a reminder of what we already know, that rare diseases are not so rare. They affect people of all ages, and that there's significant unmet clinical need for development of therapeutics. So as we know, genetics involve the flow of information from DNA, our genetic blueprint, to RNA, which provides information to translate to protein, which is primarily responsible for function. So disease-causing genetics begin at the DNA level, such that individual mutations or variants, such as SNPs or single nucleotide polymorphisms, can be incorporated then into RNA. And that could also influence RNA expression. And then this produces disease-causing proteins that can either be decreased or increased or decreased. So we have typical traditional therapeutics, such as small molecules and antibodies that target directly the disease-causing proteins themselves and either increase or decrease the function of specific disease-causing proteins. So we now have the capability to target at all levels of this genetic flow of information. So we can now target from DNA through RNA through protein. And so using modalities such as CRISPR or gene therapy, we can edit specific DNA bases or remove or insert genes or introduce a healthy sequence using cells' endogenous machinery. In addition, we can also modulate RNA expression. So either by reducing protein production of a specific toxic protein with modalities using specific designs such as antisense oligonucleotides or siRNAs, short interfering RNAs. RNA can also be targeted to increase protein production in diseases that have a deficiency. And this can be done using mRNA as well as antisense oligonucleotides. So mRNA directly delivers a healthy sequence to the cells, and ASOs use a mechanism such as altering splicing to remove a toxic protein. So this is a very busy slide, but I wanted to include it for reference as it nicely visualizes differences between modalities, at least for traditional approaches and RNA targeting approaches based on structure, mechanism of action, size, so molecular weight, and how they're chemically modified, the routes of administration used, and the approaches for delivery. So one example, ASOs shown here are single-stranded, chemically modified RNA-DNA hybrids. They target RNA through Watson-Crick-based pairing and can be designed to, as I mentioned, increase protein production through modulation of splicing or decrease protein production through an endogenous enzyme-mediated mechanism called RNase H1. They can be delivered either naked or they can be conjugated, and this assists in distribution to specific tissues related to the disease of interest. So now what I wanted to do is share the inspiring story of Susanna and her family with you, as her experiences motivate me every day. As usual, she had a very long, though unfortunately typical diagnostic journey before her parents found the N-Lorem Foundation. Her condition led her to have significant pain, limiting her capability to speak, and resulted in limited mobility, and she was wheelchair bound. This video starts with her parents describing how challenges Susanna had were transformed with the treatment and led them to their new what is. So this is very short, but it's very meaningful for me, so I want to share it. I just wanted to share this with you because Susanna is just such a special person that we were able to create a personalized therapy for her that took her from being wheelchair bound to, and her parents concerned that she had to use her own hands to move her legs, to being able to swim assisted and move her legs independently. And so for me, it's just a very motivational experience that I wanted to share. So I will. Okay, so then I wanted to share some of the process that we used at N-Lorem, really describing the complexity of developing these personalized N-of-1 treatments. So this involved really extensive collaboration between patients, families, physicians, treating institutions, as well as multiple and internal, external experts. And so this was required for each individual patient to understand the relationship between the genotype and the phenotype, and whether there was a means to administratively treat the patient for life. So each application was evaluated deeply to understand how the identified mutation for the variant affected protein function and what signs and symptoms were present that corresponded to that genotype to indicate that it was a single mutation that was responsible, and whether an ASO approach is amenable. And as I mentioned, we also had to make sure that the physician's experience and understanding of the disease, as well as their corresponding institution, was able to support administration of the therapy for the rest of her life. So I then wanted to talk about how more and more approvals for ultra-rare diseases are happening each year. So this figure in particular shows an orphan drug approval since 1987. And this is significant because the Orphan Drug Act was enacted in 1983. So it's incredible how companies really jumped on the opportunity once this act was approved to begin using this process to identify therapies. So the red line indicates FDA approvals, and the green indicates EMA approvals, which are following closely behind. And the blue line indicates an ever-increasing publications on ultra-rare diseases. And this is critical for us to understand mechanisms in disease and also to instruct companies in which modalities are appropriate for which variants, as some variants lead to a toxic gain of function, some to a loss of function, and some have aspects of both mechanisms. I also wanted to include the pie charts here on the right that show, as of 2018, that small molecules, which are shown here in blue, are still the predominant modality that's used for developing therapeutics for ultra-rare diseases, at least through the orphan process. But what's exciting is that alternate modalities are emerging. So I then wanted to talk about the typical process of drug approval. So it's very long. Typically, for this, it's shown in A here. The typical process can be between 10 to 15 years. It's very complex, and in the end results in less than 12% of drugs that reach phase 1 that achieve approval. The costs are also typically very high, reaching around, on average, $2.6 billion. So what's encouraging is that if patient-specific experimental therapies are really changing all of that, enabling a much shorter and economically viable approach. And as you've probably recently heard, that Martin Makary is the new commissioner of the FDA, and he's declared the importance of enabling development of drugs for rare diseases, based on mechanistic understanding. So the future is looking really positive for N of one, and then also looking forward to the N of many for ultra-rare disease therapeutics. So I then wanted to share, I'm not going to read through all of these, but wanted to share some of the companies that are focusing on pursuing therapeutics for orphan and ultra-rare diseases. So as you can see in this table, this is for some for-profit companies. There are so many, this is not an exhaustive list, but you can see that these companies are pursuing all modalities that we discussed above, from traditional approaches, small molecules and antibodies, to gene therapies and ASOs. And these companies span from the US throughout Europe. And when looking at the next slide, this is now for nonprofits, and you can see the same case from the US to Europe, and using multiple therapeutic modalities from gene therapies and through ASOs and small molecules. I then wanted to show this slide as well to show that there are many companies that are also looking into repurposing therapies. This has a likelihood of moving faster through the clinic, as they've already shown safety, at least in one therapeutic area or one indication. So I just want to mention again, these are not exhaustive lists. You probably know lots of other companies that weren't even mentioned here that are doing amazing things for patients in this ultra-rare disease space. So at this point, I just want to say just how very exciting this is. Time for development of ultra-rare disease-modifying therapeutics that are targeting all stages of the flow of genetic information to ultimately develop the right drug for the right patient.